Good morning, everybody. After the topic of augmentative biocontrol that we heard yesterday, where the business is pretty clear, you have products that you use for biocontrol, quite similar, as was mentioned, to what you would do in other control methods like chemical control. You have a problem and then you release. Here we are talking about conservation biocontrol, so it's a different approach. The approach is using what's already there in your system and trying to stimulate those biocontrol agents. Conservation biocontrol can be combined with augmentative biocontrol, so you can release organisms and then try to maintain them in your system to avoid that you have to release on a weekly basis. I'm very happy that we have a number of speakers today here who will cover the different bases of conservation biocontrol. I will introduce them to you later on. And I will start off with giving you an overview of what conservation biocontrol is. Okay. Can you hear me better this way? Not too loud? Okay. Right. How can I fast forward the slides? Oh, it's there? Oh, yeah, I see. All right, if we look at natural ecosystems, then we will see that, or you see that there's very rarely any pest or disease outbreaks as we know them from agricultural systems. And that is because in a natural ecosystem you have this large biodiversity and the different trophic levels that control each other and keep the system in a more or less stable equilibrium. An agricultural ecosystem, as you know, looks quite different from a natural ecosystem, and two aspects that are important when it comes to pest outbreaks is that there's a lack of landscape structures, so it's really these monocultures that we're talking about, and the fact that in agricultural systems they often rely heavily on pesticides, and the pesticides kill the pests, but they also kill the natural enemies that normally would control the pests. So you generate a system which creates continuous pest outbreaks and thereby continuous reliance on pesticides. If we want to stimulate the natural enemies that actually can help us control the pests, then we have to look at what are the resource requirements that these natural enemies have. And well, the resource requirement that we think of is they need prey. That's why we use them in biocontrol. But they need more than just prey. They also need non-prey food, which is something that people often are not aware of. They need overwintering sites, they need shelter, and they sometimes need overposition sites. And these resources are typically not available in your agricultural monoculture. So we can look at what are these resource requirements and then try to provide them in one or another way. What I mentioned, the fact that these biocontrol agents are known for their interactions with the pests, but what people don't know usually is that they also require floral resources, being nectar and pollen. And in the absence of these floral resources, these biocontrol agents, let's go back here, don't function. So, for instance, here we have a hoverfly feeding on the pollen. Hoverflies, they will not oviposit, lay any eggs if they don't have pollen to get protein sources from. These adult hoverflies don't feed on aphids, it's the larvae. But if the adult can't lay eggs, obviously biocontrol doesn't work. So we need to provide these sources. Nectar is another important food source as well. And this is an overview that we made a while ago about different groups of biocontrol agents and their reliance on floral resources, and you see that basically everything that is being relevant in biocontrol is on this list because all of them require nectar or pollen feeding. So it's something that we should not overlook. So if we want to provide these resources, how can we do this? We can either provide them through the crop itself by selecting for certain cultivars that have more of these resources. We can provide the resources through the landscape by, for instance, including floral flower strips in the landscapes, or we can provide these resources also through supplementation, so by spraying the resources on the crop. An example of providing resources through the crop is these extra floral nectaries. Those are nectar sources that are present on the foliage of certain plants. Not all plants, but quite a few plants have these nectar glands on the leaves or other non-flower structures. And the function of these nectar glands is actually to recruit biocontrol agents. So nature uses the nectar to stimulate biocontrol agents. And in a number of major crops, we do have these extra floral nectaries. So if these extra floral nectaries are present, you can actually select for cultivars that produce more of this nectar. Examples of these crops, cherries, prunus species, vicia, zucchini, roses, cotton. Those are all crops that have these extra floral nectaries. So is there a business model for plant breeders and nurseries to include biocontrol in their breeding efforts? Then providing resources through the landscape, that's these flower margins that can be used, and we'll have a nice presentation. The next presentation will go into this in more detail, or next two presentations actually. And these plant species in these flower mixes can be selected so that they are particularly suited for biocontrol agents. It's important to know that not all flowers and not all flower mixes that are on the market are suitable for biocontrol agents, especially the ones that are selected for pollinators tend to be not suitable for biocontrol agents. So there is a business model here for seed companies and for advisory services. To show you here that not all flowers are suited, in a review that I did with Paul van Rijn in 2012, where we looked at a number of flowering plant species, 3 groups of biocontrol agents, the hoverflies, lacewings, and parasitic wasps. And then we looked at which of these flower species were being exploited by these 3 groups. And you see that the lacewings, they can exploit most of the flowering species, less so in the case of the hoverflies, and the parasitic wasps were really dependent on the ones that had the most accessible nectaries. So if you want to create a flower mix for the purpose of stimulating biocontrol, conservation biocontrol, you would actually go and take the flowers on top, which are suitable for all major groups of biocontrol agents. Yes, we've done this work on these floral resources for almost 30 years by now. In this period, we have initiated a number of projects throughout Europe. So projects in the UK, Switzerland, where I started, then the Netherlands, Belgium, and Germany. And we're lucky that we have somebody from the Netherlands, which is actually the longest running project in this respect, to present the case there. Then the other approach that you could take, rather than having these beautiful flower strips, not all farmers are necessarily able or willing to use these. There's also the possibility to supplement the food by spraying the food onto the crop. And there you can have different approaches. You can actually provide those food sources that are provided by flowers, meaning pollen up here, that's commercially available. You can provide sugars as a representation of nectar, but you can also apply fictitious spray, so alternative spray for the biocontrol agents. And there's a number of products available here, Ephestia, Artemia species, where we'll have a presentation on the Artemia later on as well, and prey mites. And lastly, you can also actually produce an artificial diet, which can be sprayed on the crop to achieve the same effect. So this represents a range of different business models. To give you an example that we developed ourselves is the pollen, where we used pollen of Tifa, which is a plant species that occurs worldwide, can be easily collected, and this Tifa pollen can then be applied into the crop. This is how it would look like. The pollen is then on the crop, and predatory mites in this case can feed on the pollen. They can build up large populations on this pollen without there being any pest present in the crop. So you can actually build up what is called a standing army using these food supplements, and once the pest arrives, the predator is already present, which means the pest usually doesn't do any damage. Here an interesting approach that I tried out last year. It's an onion crop in Belgium. In the back you see the harbor of Antwerp. And this is an onion crop by a conventional grower who sprays 4 to 5 times against thrips every year. And I asked him in a corner of his crop not to spray at all. Instead, I used this pollen that I blew onto this corner of the crop to stimulate the natural enemies that are already present. And this is then how the crop looked like at the end of the season. What you see in brown is the conventional part of the crop, which has been sprayed 4 times with insecticides, completely damaged by the thrips. And the corner here in front has not been sprayed at all, and it has only been treated with the pollen. And this shows you that, and what was said yesterday, that biocontrol is less effective than chemical control. Not necessarily. I mean, biocontrol can be much more effective as well. And we also assessed then the yield of this crop. We were able to show that he had 15% higher yield on the corner that was not treated as compared to the treated corner. Sugar is another thing that we can look at. I will not go into detail given the fact that I'm running out of time, but we've done trials in citrus in Spain where we were able to show that by the use of sugars you can actually, in combination with the release of parasitic wasps, you can raise parasitism levels, double them, and the parasitoids also are maintained in the crop for a much longer period of time. So here you see that you have a much higher parasitism. Prey mites being used on the crop, that's another approach that has been shown to be quite effective. Again, on the left, no prey mites. On the right, you see predatory mite levels with the use of prey mites. Food is one example that I've covered now with a number of cases, but you can also think of other resources. Domatia, which are these hairs that you find on a number of crop plants, is another one, and these domatia that you see here, you see that there's little balls in there. Those are predatory mites that are sheltering in these domatia, and they also use them to lay their eggs. So you can either select for crops which have more of these hairs, or you can also apply these hairs artificially. Here you see the eggs being laid on the Nomatia. Well, there's an issue that not all crops have these hairs, so a number of important crops are actually hairless, and in those cases you can stimulate biocontrol by providing these hairs artificially. So here we have done some trials where we applied different combinations, sugar, pollen, fibers, and then looked at the impact on predatory mite levels. If you apply one of these resources by itself, the impact is fairly limited. There's a little increase relative to the control, but not much. If you combine the two, and especially if there's fibers included, you can see that the numbers go up considerably, and if you compare them or combine them all three, you can reach really high levels of biocontrol agents. So this shows you that there are synergies to be had here, that 1 + 1 is not 2, but 1 + 1 in this case is actually 20. And I hope that I stayed within the time. Thank you.
Thank you very much. Do you have questions for Felix? We got time for 2 or 3 questions. And Violette, don't forget to say your name for the people in
okay,
distance.
wait, thank you. So thank you for the presentation. Anne-Violette Lavoie from the University Nice-Côte d'Azur. I have a question regarding the food supplementation and the example with the predatory mites and the pollen. If there is a preference for the predatory mites between pollen and prey, because actually they are looking for nitrogen for both, no?
Very good question. You could imagine that you get a satiation effect. And predatory mites that have eaten a lot of pollen will not be as motivated to eat prey. And that is indeed something you find if you look at the short term. But given the fact that this pollen supplementation leads to much higher reproduction, already in the next generation, this effect turns around. So and given the fact that this predatory mite population time is about a week, so the effect is really very short term, and the benefit is over the rest of the season.
There is another question by Marc.
Thank you, Felix, for your talk. Sorry, Marc Bardin from INRA Avignon. INRA et Avignon, sorry. How to be sure that nutrient supplementation does not promote the development of pests or even pathogens on the plant?
Well, the pathogens is a question that is rightfully raised, especially in the case of sugar sprays. And we all know that if you have sugar levels on your crop, that you get sooty mold growing on it. Other pathogens can benefit from it as well. So the trick there lies into the concentration and the way it's applied. So by going down with the concentration at low concentration levels, one, you avoid that your whole crop becomes sticky, which is something the farmer wouldn't like to see either. And you can also then spray it in a very fine mist so that only a few little droplets get onto the leaf. And under those kind of conditions, there is no issue with pathogen or sooty mold growing. And at the same time, the targets that you're looking to feed will still find these minute droplets and be able to feed from them.
Last question by Simon.
Hi, Simon Felus from INRA. Felix, so the picture and the graphs you presented were very convincing, but I imagine like for anything that in the real life, there are cases it works, cases it doesn't. And so my question is, in practical terms, do you have recommendation of for example, surveys to carry out before deciding to deploy or not to deploy these feeding techniques? For example, do you think the farmers need to look around, see whether they have the predators or not, and then decide whether to feed them or not, or what's the rationale there?
That's a good point. I just want to make clear that these are all examples that also have been used at commercial farms. So it's not just a laboratory artifact that we're presenting here. The preventative use of this conservation biocontrol requires a different mindset and a different approach from the side of the farmers. And I agree with your point that you made, that you would like to know what is there already in order to see, can you actually stimulate it? Because if you start with nothing, you can put out these food sources, but it will not have an impact. So an approach that we are also looking at is to do some monitoring, in this case monitoring for your beneficials rather than monitoring for pests. And on the basis of this monitoring, it will tell you what your natural enemy population is. It also allows you then to, if you are considering releases, to do targeted releases. So if you see, for instance, in the case of aphid predators, there is a lot of variation from our experience from year to year. One year it's ladybirds that are doing the biocontrol. The next year it's hoverflies and so on. So if you do this kind of screening beforehand, you see which key biocontrol agents are missing, and then you can do actually your releases in a targeted way, that you release those that are not present, and that you don't end up releasing something which is already in the system anyway, because that would be a waste of money for the growers.
So this service, it's something that you recommend, that's something that is done, or is it for the future?
It's something for the very near future. We will have a presentation by Rob Moerkens later on, showing how we already developed this for greenhouse systems. And so it's a small step from the greenhouse system to an open field system. And it's, I think, going to be a big step forward for biocontrol and especially for conservation biocontrol.
Thank you so much, Felix.